Thank you, Anthony, and my thanks to the Garvan Institute for this opportunity to talk to you today, sort of in the long view of where we've come over 200 years, taking the milestone mark of Sir James Parkinson's original description, and how far we've come in the last 200 years, sort of set the stage of where we are and where we can go, really in just the next few years. As mentioned, I'm an investigator at Massachusetts General Hospital in Boston. I actually run a Parkinson's disease research laboratory on the Navy Yard campus in Boston of Mass General Hospital, where the USS Constitution Ironsides, Old Ironsides as it's known, is berthed. And we're just across the Charles River from the main campus and linked by what's now about 15 years completed, the Anzac Bridge, sort of the crown of what was notoriously referred to as the Big Dig for a number of decades, and a bridge that we've adopted initially for some of the work and some of the molecules we've been targeting Parkinson's disease over the last couple of decades, and the idea of taking the understanding insights about the disease and particular promising candidates and going across a bridge from the laboratory to clinical therapeutics. And so that's a theme that you'll see is really what marks the centuries that I'll try to span here, but also with a friend on beautiful Sydney Harbor this weekend that we got to enjoy, realized that the metaphor for that translational research extends very nicely to what had caught my eye, which is the Anzac Bridge, the Australian-New Zealand Army Corps, I think a commemoration, actually World War I contributions that so beautifully laid out in its very parallel architecture. So as you'll see, with the Internet's availability of a lot of beautiful images, I'll bring you back to the Anzac Bridge as well. So what I'd like to cover in the next 20 minutes really is just after a very short thumbnail of the clinical picture, which really is, many of you are much more the expert than I, and was already so nicely summarized by Clyde. And then I'll give you sort of that timeline, bird's eye view of where we've come and marking advances that lead up today. And then really the last few minutes, I just wanted to highlight the opportunity to work all together and the many contributors to the prospects of building on all that's been achieved in Parkinson's research for the last epoch. So just really in one slide, already, again, much more aptly presented the core clinical features, the spilled pint, much better visual than just noting tremor at rest in amongst the classical features of Parkinson's disease, the classical motor features. Parkinson's is thought of and grouped as a movement disorder, really more of a lack of movement disorder with slowness and stiffness of the muscles, is sort of this classic F. Netter depiction captured here without video. Again, the only real extra movement, short of some of the side effects of medication, is a rhythmic tremor, notably at rest. There are many features that get wrapped up in these sort of cardinal symptoms and signs, but these are what are encapsulated in sort of the constellation that describes Parkinsonism, usually caused by Parkinson's disease. As it progresses, as it inexorably does, although to a very variable extent over, not rapidly over hours or weeks, but over years and decades, there's more disability in the movement sphere that's most manifest as walking difficulty, balance difficulty. But increasingly, as I'll highlight amongst the discoveries in the recent decades, there's been an appreciation of what most people who've been living with Parkinson's learn the hard way themselves, is that it's more than just a movement difficulty, but they're so-called non-motor features, and both in the central nervous system as well as the involuntary nervous system, and faculties as well becoming involved as the disease progresses. Okay, so for that bird's eye view, what I'd just like to go through in sort of a timeline is certain, since we're going to reach all the way back to the original research, if you will, which was basically a description by Sir James Parkinson, over 200 years ago, and then what led up to sort of the description, not just in how people who are dealing with disease appear in the clinic, or really in his case on the street as he observed them in London, but also as we've observed more closely at the brain, what parts are involved, what chemical signals, and then with the more recent research that beyond the classical dopamine chemical signal that's been implicated, how we've gotten a broader view of just describing the disease. And then I'll touch on the advances that have come in the way of clues about genetics and environmental potential influences on the likelihood of getting the disease, and how that knowledge has led us to identify at a molecular level potential candidate targets, and then drugs that can meet those targets to try to slow down the disease. I will focus, although I'll highlight the history of the paradoxically very welcome and effective symptomatic therapies that we have for the disease, in contrast to some of those other conditions that Clyde mentioned, for which we, like ALS, for which we have no substantive symptomatic treatments. How we have those, yet those aren't adequate, and on top of that, the underlying cause of the disease, the progression of the disease, we don't even have any validated treatments for that. So in moving through how these advances have led us towards the prospect of gaining those kinds of therapies, I'll also finish on the distal end of how those candidates that come from all this building knowledge get validated in the laboratory, leading to candidates that are identified and ultimately get tested in clinical trials. So this is just sort of a timeline representation of the last 200 years or plus, starting with that seminal descriptive contribution. Each one of these little yellow spots is intended to indicate a major advance or breakthrough in our understanding of the disease. And one of the points I wanted to illustrate with this timeline is how these advances have really been accelerating to the point where it's almost a crescendo now, and as I'll try to only partially go through, just in the last 20 years, the pace at which those advances have been coming. But going back to 200 years ago today, this fabled monograph, the essay on the shaking palsy, was published, and it was, as mentioned, still sort of classical and medical literature for so incisively identifying the features that I mentioned, descriptively observed by Dr. Parkinson. So it's a long gap before, as you can see, before we began to to move into an understanding beyond just simply description. And about 100 years later, it was figured out the part of the brain that seems to be, at least for the classical motor features, involved in their manifestation. And just to take you back to beyond the clinical features, that identification pathologically, which is basically looking at the brain in parts, not even under a microscope, but just this is a section at the top of the brainstem that is sort of a cutaway section from someone who died healthy or without Parkinson's disease. You can see this black stripe, which is named the substantia nigra, for a black substance, and reflects the black pigment in the brain cells or neurons that degenerate prematurely in Parkinson's, such that this difference was seen about 100 years ago, that this region seems to be involved because of the blanching or the loss of the black substance, reflecting the degeneration of those pigmented brain cells or neurons. Here's that black substance here at the top of the brainstem. Here's the spinal cord, brainstem, the hemispheres of the brain over top. So at the very top of the brainstem, you can see represented here, if you kind of slip the brain back in, those black stripes represent a cluster of brain cells that send out their axons or processes higher up into the brain, into another cluster of brain cells in a sort of a circuit that where they release a chemical substance that facilitates normal movement. And in Parkinson's disease, as mentioned, we lose those, people who have Parkinson's lose those brain cells early, so they lose some of the innervation to that structure, and they lose some of the facilitation of normal movement. And that's where we were about 100 years ago. next huge advance came with the identification of what that substance releases, the chemical signal that gets released by those rising neurons into that circuit as dopamine, as a neurotransmitter, which was then found to be at markedly lower levels in the brains and the substantia nigra and the target region of people who died with Parkinson's disease. That led in fairly short order to really what is the gold standard and very effective, as I mentioned, symptomatic therapy that we have with the establishment of levodopa, which is not dopamine itself, but it's a precursor that the brain can take up and convert into dopamine so that on its own terms, for a time, some of the lost movement in Parkinson's can be regained. And really, typically, even though the disease progresses, tends to be effective for the duration of Parkinson's. Then, prior to this last couple decades, a milestone that began a discussion of the influences on Parkinson's disease, whether there was something in the environment that may be triggering them, or a genetic composition. The first salvo in sort of that back and forth and ultimately sort of a integration of genetic and environmental factors was the identification of a neurotoxin called MPTP by some very talented medical detectives in California with an outbreak of what seemed to be in young people, Parkinson's disease, amongst drug addicts who were taking something that was traced to a bad batch of heroin-like analog that had the substance MPTP that could then be given to laboratory animals and recreate Parkinsonism. And it was taken as actually a great observation and clue that led to a new way to look into Parkinson's, to model Parkinson's in the laboratory. But it also highlighted the concept that there are things in the environment, at least in this small subset of people, that could be influencing the risk, if not causing Parkinson's disease, although it's certainly not. Those were the exception, which was also the case for what marks the last 20 years. So I won't take you through every little dot, and I'm only semi-randomly highlighting some of the most salient advances. But one of the features that have really rocketed us towards much greater insight, in addition to, as I'll show you, incremental insights into potential environmental factors, there's a series of genetic clues that started with the reporting 20 years ago this year of the first of a number of genes that have been implicated, the blueprint material for our bodies, the genetics that has led to considerable insight. So the first one, a gene for something called alpha-synuclein, was identified then and began with really, it was called Park1. Park2 was, instead of a dominantly directly inherited, was a so-called recessive or pattern of inheritance for a family form of Parkinson's that it's really rather uncommon, but these were huge clues because in these cases we were learning something about, at least in some people who have Parkinson's, something that could actually cause the disease. And so without going into the detail, this has sort of led to a revolution initially in terms of individual genes, other ones called LRRK2 and GBA, still only a small percentage of the genetic contribution to Parkinson's, but incrementally we were getting a handle on some of the biology that's given us a clearer but still early picture of Parkinson's disease. Some of those genetic clues have manifested in the form of other advances like stem cell therapies, which are still early, but are additional insights that we can build on. I just wanted to highlight in this next series over the last 20 years a number of complementary clues that have highlighted, despite the importance and value of these genetic discoveries, that as was reflected seminally in a paper of twins on a large group of World War II twins, comparing twins who one of whom has Parkinson's to whether the other twin has Parkinson's, these large cohorts, and then seeing what the likelihood of that other twin having Parkinson's is as a function of whether that's an identical twin or a fraternal twin. And as it turns out, for most people over 50, the likelihood that your twin, if one had Parkinson's and you got it when you were over 50, the likelihood that your twin has Parkinson's was no different if your twin was identical or fraternal, suggesting that genetics was not the key to typical Parkinson's. But actually, if you got Parkinson's before you were age 50, there was a significant greater chance that your twin would have Parkinson's if it was a genetic twin, highlighting that genetics plays an important role, particularly early in the disease, and as we learn and more subsequently at a lower level, genetics, very tiny genetic influences that you can't tease out with the kind of studies I'm showing here, suggest that many genes are involved in the disease, and actually that's the kind of work that I know Anthony and his group here at the Garvan is making advances in parsing out. The last sort of set of milestones before things get really crazy here, I just wanted to highlight was that we're continuing to expand our understanding of what Parkinson's is, and these insights are also helping us develop improved therapies. So I just wanted to highlight another advance that's often where we've begun to think about Parkinson's in a different way. A neuropathologist in Germany, Heiko Brock and his team, identified that the product of that gene discovered back here as a clue that suggested it wasn't just those dopamine-producing brain cells at the top of the brainstem and their degeneration that contributes to the disease, but it actually is much broader disease, which fits with those non-motor features that I mentioned and suggests that other brain regions are involved and even regions outside the brain in this theory that's being actively tested. I just wanted to highlight lastly here in this timeline what I've considered to be one of the more instructive advances that came right out of Sydney, the Sydney multi-site cohort that followed patients with Parkinson's for decades and highlighted the fact that in this publication a decade ago that the majority of people with Parkinson's end up really struggling less with the motor features and more these other non-motor cognitive and related conditions that fit with our broadening understanding. So it's become dizzying in some sense and exciting in the last 10 years how all this knowledge about the risk factors and the biology is leading to candidates that, for different kinds of drugs that we can now begin to prioritize and begin testing more systematically in the clinic. Just in terms of the relationship to genes and environment, this is, I feel, often a little more dramatic, but it's still instructive that genes sort of set the stage or load the gun and environment pulls the trigger, and the idea is finding what target we can that will stop that process. So I'm not going to go into any details about any of the dozens of leading candidates. I just wanted to highlight in explaining a little bit how we think about how these clues have helped us understand the causes and the mechanisms of the disease. Just point out in this genes and environment dichotomy that in both cases, for example, for genes, we have a number of genes, as I was suggesting, actually can, generally in very small families and cohorts, be causative. And then there are many more genes that we're understanding, particularly from the genomics approach of Anthony and colleagues, that are risk factors that influence the likelihood of getting the disease or that it will progress. And then environmental factors, similarly, very rarely can they be causes, like those MPTP addicts, and more likely that they are risk factors that modify the risk. And that leads, in our way of thinking about it, how these triggers can then lead to the generation of brain cells, like those dopamine-producing brain cells. And there are many theories of what the actual processes are. I won't go through these. I'll just highlight one that is still central and is supported by both genetics and environment, that oxidative damage plays a role in Parkinson's disease. So here's that thin section right through the middle of the brain, at the top of the brainstem, that shows, again, the substantia nigra lost in a patient with Parkinson's disease, and frames this process amongst the different potential ways in which this can happen as an oxidative pathway where metals, which are also thought to be tied up in the pathophysiology, and oxidative damage lead to the degeneration. And much maybe like a vintage car from when some of us were first born, they can be fine machines, but with time they can break down, and part of the process can be similarly oxidative damage. And the idea here is to find, for this particular target, how can we slow this process down, just as metaphorically it can be done with a good coat of good chemical substance. You know, what is the Rust-Oleum here? And there are a number of candidates, whether it's this process or related processes, that have come out of all these research advances and brought us to this point. where we are now trying to sort through these processes and the molecules that are suggested that are at play, and the molecules that can be the drugs or the other treatments that can keep them from progressing. And that's why, I mean, it's maybe a little overstated as a tip of the spear here, but the metaphor works, it seems to me, is we're at this point where we're learning so much so fast, we're lining up compounds, and really the next few years, I think pushing forward here, building on these advances, is a very hopeful time, but as in the last few minutes here I'll try to highlight, is something that really is much more likely to happen and happen sooner, working collaboratively. So working or walking the bridge a little bit further here, I'll just highlight the different roles, including the public and people with Parkinson's is well represented here. It's kind of buoyed by the increasing connection I see for what goes on down here, having met with the clinical scientists last week, as Clyde was mentioning here, in the Garvan-hosted collaborative effort with the Parkinson specialists who are interested in coming together for this effort. You know, the foundation, first of all, in terms of the many people to come together, here's the A-frame of the South Tower in the Anzac Bridge across Boston Harbor. And here, off the internet, is the A-frame coming up for the Anzac Bridge. And again, I think the bench scientists who really understand the biology, like Antonia's group, are fit right into that metaphor. And then, you know, building on that, the platform gets built out. And here is which was the focus that the Garvan and Link Clinical Trials has brought us together to focus on whether the infrastructure of the clinical trialists are down under here can be brought into onto the bridge, if you will. And here I just wanted to highlight, without going into much detail, that for decades now in our corner of the globe, we've been pursuing on the clinical trials end through a group called the Parkinson Study Group that I lead now, that is the largest nonprofit for group of clinical trial sites, and the trialists and the coordinators and allied professionals who work together to actually test these leading candidates. And been doing it for the last 30 years, but as I say, and this is the sites in North America where we're located. Although we've been working on a lot of compounds, the ones highlighted are actually more on the symptomatic end, we've been increasingly conducting clinical trials geared towards slowing down the disease. And so we've worked out an approach and trial strategies, science in and of itself, that for what trials have been done in North America, working with the Link Clinical Trials group and others, and we're excited about the prospect of working really with individuals. These are the individuals of the original large-scale clinical trial for disease modification that convened back in the 80s, wearing on their T-shirts Datatop, which was the first major clinical trial for neuroprotective candidates globally, and how it's the people that have to come together on the clinical trial stage. And so it was one reason why I was so excited to see not only the expertise of the clinical trialists and the clinicians here, but their enthusiasm last week for joining this. So here I'm just highlighting another integral foundational component, which is the funding. And again, it's with all the work that's gone so far, in some sense, I find these metaphors to be most compelling when the bridge is just about complete here, and that was the case for the Anzac Bridge, as was so nicely mentioned with the people motivation of not just people with Parkinson's, but their families who motivate them in Clyde's compelling description of the kinds of advocacy groups like Shake It Up Australia or the Cure Parkinson's Trust, hopefully taking us across the bridge, the Fox Foundation, and a number of other foundations. You're going to hear more about the Link Clinical Trials Initiative, which is that of the Cure Parkinson's Trust, bringing together scientists and people with Parkinson's, like its founder, Tom Mozix, and represented by Richard Wise, the scientific officer, to try to not only advocate, but actually take up, in the case of this Link Clinical Trials Initiative, this last step of finding the best compounds and bringing them to trial. And so lastly, I just wanted to acknowledge, thank, and highlight that the people with Parkinson, people who are dealing with Parkinson's, are really arguably the strength of this whole endeavor in this bridge. And I thought that these remarkably similar cable stay artistic renditions here, both the Anzac Bridge and here the Zakim Bridge, really represent sort of the steel of the metaphor in making this happen. And most obviously, these trials can't be done unless people with Parkinson's volunteer to test and be equally likely in a good trial often to be in a placebo as well as a candidate drug. But the additional role, as Clyde was highlighting, becoming advocates to make this happen, not only just to bring volunteers to the trials, but also to motivate the players, the scientists, and the funders to put something on the table to get across the bridge. So that's what I've just tried to highlight here, that we've come a tremendously long way in the last 200 years, and the pace is just building, and I appreciate and have very much appreciated in our chance to visit beautiful Sydney, that this metaphor is really very apt, and it's a very good time to work together over this prospect. So thanks for